Hello, everyone. Thank you for joining our webinar today titled Rapid Microbiological Methods. I'm excited to have Dan DeMarco, Doug Marshall, and David Legan here with me today hosting the webinar. I am Amy Knock, and I will be moderating today's session. Before we begin, I wanted to let you know more about how this webinar will run. The webinar is being recorded, and the slides and recording will be available to you within 3 business days. A short Q&A session will follow the presentation to answer any viewer-submitted questions. During the webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your questions, then enter the Enter keyboard on your keyboard. Remember, you can submit questions throughout the webinar. That concludes my brief introduction today. I will pass it over to our presenters.
Thank you, Amy. I think I'm on first. I'm David Legan, Director of Science out of our Eurofins Microbiology Laboratory in Madison, Wisconsin. And thank you to those of you that completed our survey. Next slide, please, Doug. Just thought we'd very briefly run through that because sets a little context. In the audience today, we have a wide mix of people, senior professionals, mostly in quality assurance, maintenance, sanitation, R&D, and microbiology, not surprising. We also have some sales and marketing people, welcome. And very clearly from the number of responses, many of you wear multiple hats. Your organizations, about 2/3 currently are not using rapid methods, which I guess is why you're here. Of the third that are, you're using a mix of technologies and mostly using either central corporate labs or third-party labs. You generally trust your rapid methods, which is good. And the 4 most significant things that you test for safety are coliforms as indicators, E. coli, salmonella, and listeria. About 1/3 of you said you're doing confirmation of pathogen results. I suspect that ties to the 1/3 that are doing rapid methods, and you're doing those at an off-site location for the most part. You said that you wanted methods that are faster than you have today and more sensitive. Those are your 2 primary asks. As we go through the webinar, you'll learn that those 2 wishes really work against one another. So we'll explore that. And specific questions that you raise that you'd like to talk about are false positives and validations, and we'll spend some time on both of those things. So over to you, Dan, to get the technology started.
Thanks, David. I appreciate that. I'm Dan DeMarco. I'm based out of Louisville. I'm actually coming to you from Louisville today. I apologize. I'm not sitting in a dark room, if that's what you're thinking. We had some technical difficulties this morning getting set up, and so actually Doug is going to be driving the slides for me. There's a lot of clicks in here, so if it gets a little bit awkward, we can blame Doug for that. But essentially, when I was charged with pulling together this talk, I was given this kind of task. They said we want an overview of rapid methods in food pathogen testing. We want to include the biology and chemistry detection, and we want this to be done in 10 to 15 minutes. And this is really an impossible task. I mean, the field is complex. It's been getting more complex every year in the 25+ years that I've been involved in it. If you could just advance. Go ahead, Doug, click, please. And oh yeah, they also wanted me to try and squeeze in some terminology stuff as well. So to give you a sense of just how complex this is a table I tried to pull together, basically summarizing what I consider the major methods and categories of methods in food pathogen diagnostics. And you sort of slice and dice the field in all sorts of different ways. This table is a summary, a condensed view of the actual table, which is actually quite a bit larger than this. But in any case, I broke this down basically into the basic major categories of detection technologies, which I considered the affinity ligand technologies, molecular sort of amplification-based technologies, and then sort of hybrids and others. And if you could click, Doug. I don't want to go into too much detail here. I recognize this is pretty much an eye chart. You will have the slides afterwards because there's a lot of information here, but I just want to highlight a couple things. And click again, Doug, if you don't mind. So within the affinity ligand category, so these are technologies that are based on the capture of a specific target using an antibody or some other ligand, like say an aptamer or phage protein, etc. You have lateral flow and dipsticks in here, and you have your ELISAs and alphas in here as well. If you click again. And then in the molecular technologies, of course, you have all the PCRs. And then, of course, these are very complex in and of themselves, and an entire talk could be done just going into the different types of PCR that are available. Click again. And then sort of in the hybrids and others, there's only really one here that I mentioned, which is immunoPCR. And go ahead and click again. And then one more time. And then finally, sort of the want to highlight just the last couple, which is culture, which is still obviously widely used, still a widely used detection technology, although not necessarily considered rapid. Biosensors, I mentioned that because these have been made a big splash many years ago. They've been on the scene for a while, but currently, as far as I'm aware, there's not really a major player in food pathogen diagnostics in the biosensor field. Bacteriophage-based methods and sequence-based methods are also very much up and comers, and I think you're going to see a lot more from those in coming years. Go ahead and click again, Doug, and then one more time too. So this is a slide, with all those technologies, that have been brought to bear over the past 25 years. Why? First of all, why is it so complex? And second of all, why are we still pretty much where we were 25 years ago? If you look at sort of turnaround times and that sort of thing, we haven't really made as much progress as you think perhaps we might have. And one of the reasons is that what we're trying to do is really, really very, very difficult. This is a slide that I've shown many, many times over the past 20 years or so, because I really, really like it because it highlights the problem, I think, in a nutshell. What you're looking at on the left there is a 375 gram ground beef sample. That's the actual food. Then that's in a stomach bag with some enrichment media. And there's an environmental sponge. And basically, we're saying you've got to get that single target organism out of that thing and into this thing, which is a small plate or whatever your detection modality might be. And the challenge is compounded because bacteria are so small, right? So I've shown there, it's a 1 millimeter, if the screen size is right, it's 1,000x the actual size of a bacteria. And I helped David Legan and Doug Marshall help me do some math on this. And if it turns out it's a single bacterium or a 6 foot, 6 inch tall person, they would need to travel about 120 miles to make it from one side of that large ground beef sample to the other. For reference, the state of Delaware I used to reside is 96 miles long. Click again. Another way to look at this, if you think about the needle in a haystack problem, Dave Legan again help very much with the math on this, and you compare finding bacteria in a ground beef sample to finding a needle in a haystack, you do the math, and actually click again. Actually, it turns out that finding the needle in a haystack is a lot easier than finding the 1 E. coli in 375 grams of ground beef. In fact, it's 170,000 times easier to find that needle in a haystack. So just to give you a sense of how difficult the problem is. Next slide. Go ahead, one more click. So if you look at the past 30 years, and the focus really has been on detection. It's really been the main focus of innovation. And the innovations have really come, at least in food pathogen testing, from the clinical diagnostics world, and more recently from the biothreat applications. And typically the food diagnostic testing technologies are 5 to 10 years behind what you see on the clinical side. There are technologies currently that can provide accurate detection in seconds or minutes, and we can detect a single organism or molecule. You can click again. And in my view, and I've said this many times, perhaps less controversial today than when I said it 15, 20 years ago, but in my view, the detection problem has been solved. This is no longer a detection problem. It hasn't really been one for a very long time. This is really about getting the target into the detection system. So it's really a sample preparation problem. Next slide. So if you look at the current food testing paradigm and how we do sort of sample preparation and with enrichments and sampling, I mean, you've got a problem where you have to detect a single target organism in a large mass of material, and you have a small sample size that you're pulling out of that. So most all rapid methods take a very small sample size to be analyzed into the the final detection system. How do you make sure that that target is in that small sample that you take so that you actually detect it if it's there? The way we do that currently is through enrichment, and right now we have long enrichments. That's because we have this small volume that we're taking. So in order to make sure you get that one target into that 5 microliters or whatever that you pipette out of the enrichment, you've got to enrich that for a very long time to make sure the concentration is very high. Click again. The advantages of this are many. I mean, it's simple, it's effective. You can add selectivity through the addition of selective and differential agents. You can suppress competing background flora, and it dilutes out inhibitors. And it also essentially works or could be made to work for almost all matrices. Of course, the big disadvantage here is that it takes time. And again, it slows your turnaround time. Next slide. So you might say, let's sample a larger volume, which would be a good idea, and you could potentially then have a shorter enrichment time, so you wouldn't need to grow the organisms as long because you're sampling a larger volume. Hypothetically, this improves your sensitivity, and you get a shorter enrichment. The difficulties here are many. Usually what happens is the additional time or the costs involved outweigh the advantage of the sensitivity improvements. Oftentimes, it's just easier to leave the thing in the incubator. In fact, most times it is. And the difficulties increase as you increase in sample weight and as you decrease the dilution ratio. So as you have more sample to the media, the problems get even more difficult. And this might be only possible for some matrices. Each matrix might even require a unique method. And then you also have inhibition problems because now you're sampling a larger volume. Next slide. So how do you present the target actually to the detection system? Either with a small volume sample, sort of what we do classically, the kind of standard approach, or if you're sampling a larger volume. Well, basically you have two let's say high-level approaches. You can dilute that and test it, or you can concentrate it and test it. Click again. So for the small volume samples, again, sort of the classical way we do things today, it's you take the sample, a small volume, you test it directly, you dilute it basically into something, and then you test it. You can also do some sort of an extraction or purification. So this could be a physical separation, like an IMS step, or a chemical separation, like a PEG precipitation. There's all sorts of things that are done there. If you're going to test a larger volume, you have to concentrate. And again, there's different ways you can do this. You can do affinity ligand, magnetic separation, concentration. You can do centrifugation. You can do filtration. So there's lots of different ways you can kind of skin that cat too. So if you're following the way what I'm talking about here, you ask yourself the question with a standard enrichment and the standard way we're doing things today, why would you ever do things like centrifugation and filtration? And really, you wouldn't. And that's basically what you see. People aren't really doing that. Conversely, if you are sampling a larger volume, you really can't just go straight to detection with a direct approach or do some sort of just straight up extraction without concentration. You just can't, because your chances of false negatives are just too high. Next slide. So example, this is a good example of what I'm talking about, I hope. And this is an example of a commonly used real-time PCR method for salmonella detection, where you have an enriched sample of which you're taking 5 microliters into a lysis solution, 200 microliters in this case. Go ahead and click. And if you do the math on this, you will end up with 0.0243 microliters of that enrichment into that 205 microliters. Of that lysis solution, you take that through a couple heating steps. 30 microliters is taken into the actual PCR reaction. So in the end, you have 0.73 microliters of that enrichment is actually making it into the detection system at the end. That's an exceedingly small volume. Click again. So for a 25-gram sample, where you have 250 milliliters of enrichment, that's only 0.003% of the entire volume. And for a larger sample, 375-gram sample, that's only 0.00015% of the entire volume. And this is your answer in a nutshell. Click again as to why enrichments take so long. Again, you just are taking an exceedingly small amount of that original sample that's actually making it into the detection at the end. Next slide. So if you look at typical rapid methods, there are basically typically three steps to those, and 3 core components. And it's a combination of all of these 3 core components that determines the overall sensitivity and robustness of the method. And each component contributes to that sort of in different ways. And there's all kinds of factors that influence how. And so this is an example of a PCR method workflow. And I'll go ahead and click again. If you look at the enrichment step or the recovery step, whatever you want to call it, you have all sorts of things. You have the media that you use. What are the nutrients? What are the selective and differential agents? What's the matrix you're testing? What's the sample size and dilution ratio? The incubation conditions. Of course, the biology of the target organism. Gram negatives grow much faster than gram positives. Is there a secondary enrichment, or is there some sort of a regrowth step? Is it compatible with the PCR itself, the actual media that you're using or the steps you're using? And then, of course, the volume of enrichment that you transfer into that lysis reaction. Same thing for the lysis here. The media and matrix are critical. Are they effective with the lysis conditions chosen? What is the method that you're using? Is it just a crude lysis approach, or are you doing some sort of extraction and purification first, followed by lysis? And then what is the efficiency of those things? And again, are they compatible? Are those reagents compatible with the downstream detection system and the volume of the enrichment that you're actually testing? And on the detection side, again, the media and matrix play a role. What is the sensitivity of the instrument that you're using? So the fluorescence or light detection sensitivity. Most modern real-time cyclers claim to detect a single photon, but there is some variability there. What's the assay chemistry? What are the cycling parameters you're using? What are the efficiency of all those things? And the volume of the lysate that actually makes it into that PCR reaction in the end. So a common theme, I think, you click again, you'll see running through this is that volume is really critical here. And really, it is volume of sample to detection that's the primary driver of method sensitivity. All right, I'm going to shift gears big time now, and I'm going to talk about something that I actually have an entire seminar on scheduled for next week, which is non-confirming presumptives, and the great debate here about non-confirming presumptives. So I'm going to start with a true or false question for the audience, and maybe I can ask you to put your answer in the chat. And it's this: there has been a large increase in the number of presumptives that cannot be confirmed by culture in recent years. So if you just want in the chat box, I'm curious actually to see how many people say that's true and how many people say that's false. And I'll keep talking while people are putting answers in there. But I think the majority of people that I've heard from, and myself happen to believe that is a true statement. There has been a large increase. And I think as you talk to people in industry and around industry, I think that's pretty much widely understood that is in fact the case. Next slide. So if you step back in a second and ask yourself the question of why, what is a presumptive positive? What is it about a particular result that makes it presumptive versus another result that's not considered presumptive? And there's a number of reasons why you might consider something presumptive versus not. So performance is a big one. So if you have reason to question a performance characteristic of the method, So for instance, the sensitivity or specificity or its robustness, if it's not as good as the standard method, or even if it's superior to it, the gold standard method, you might want to call it a presumptive result. Or in the case of a multiplex PCR where you have 2 targets, which may be found in different organisms, you might want to call that a presumptive result. Next, click. Defensibility, this is an important one. So if you don't have full confidence in that initial result, because there might be artifacts of the analysis, so improper sample prep, lab contamination, those sorts of things, it gives you further verification of a presumptive result, the retesting there. So it's more defensible. And that's really important when you have to, regulatory actions are demanded from the results. Next. So if it's a new or unknown science, so you don't know, so the science behind the method or the target is new, it's actively evolving. So there's not enough yet known about it really, or the method really to understand how they behave in the real world. This is a great example of this is SARS-CoV-2 and SARS-CoV-2 diagnostic tests. Next, click. Misleading. So the positive result could be said to be misleading in terms of the actual risk. So for example, in the case of PCR, you can have detection of dead cells or non-culturable organisms. The presence of dead cells, DNA or RNA should present no risk from a food safety perspective. I'd argue that that's not necessarily the case, but you could say it does not. And therefore you would need a secondary method to confirm the presence of live cells. Which leads to the next important point here, which is false positives and non-confirming presumptives. And it's really critical that people understand that these two things are not the same. They really are in fact very different. And I've put together a table here to kind of hopefully help illustrate this, comparing false positives and non-confirming presumptives. And so if you look at the result that the actual instrument reports out, it's going to be a positive in both cases. The initial result that we will report out will be a presumptive, and the final confirmation result will say not detected. But the difference comes in here. In the case of a false positive, the actual target, DNA or RNA, or you can click again, or the actual organism itself is not present. In the case of a non-confirming presumptive, it is. Okay. But the causes of a false positive are cross-reactivity with a non-target organism. So that could either be primer probe specificity, or it could be some sort of affinity ligand specificity problem, or you could have a signal problem with your instrument, say, is another example of that. And the non-confirming presumptives basically are everything else. So that's detection of dead cells, that's a confirmation method failure, etc. And people tend to forget this, but it's important to remember, confirmation methods are not perfect. There's no perfect diagnostic method. Anybody that tells you that there is wrong. There is no perfect confirmation method, or no perfect diagnostic method. Confirmation methods also fail. You will have other terms sometimes for non-confirming presumptives, such as non-culturable presumptives or detected not recoverable. I think there's some other terms floating around out there, but we tend to prefer a non-confirming presumptive. Next slide. So one thing Doug's kind of flashed through it there, but it's kind of hard. Go back if you don't mind, Doug. One last point here. I think it's still true that less people understand this distinction than need to, which is one of the reasons we're talking about it. I like to talk about it a lot. For the majority of people, these two things are equivalent, and I don't overlook the fact that it doesn't really matter, from a perspective of a test user, because they have to be acted on as if they're true. That's really where the heartache comes in from this, and that's why this is such a tremendously annoying issue for so many. Next slide. So just a couple of slides, just a little bit of discussion around this. The detection problem, again, I believe was solved. It's been solved decades ago. It's a sample prep problem that's remaining, and it's difficult though. We have to find targets that are present at ultra low concentrations. Remember the needle in a haystack, in large masses of material. And these masses of material are extremely diverse. Okay, span the gamut of forms of matter, from liquids to gels to solids, the whole thing. They often have substances which are inhibitory to rapid methods, and there's a great reluctance to pay more for testing. And some of these things make it hard to make it high throughput compatible. And you've seen, and I think you're still seeing to some extent, more research dollars spent on detection than sample preparation. And the thing is, advances in detection, these have come in the past from the clinical worlds or the biothreat world. But from the clinical perspective, their lives are way easier in terms of sample prep. They have a few matrices, targets are present at high concentrations. They're basically done. They're done on sample prep, and they have been for a long time. So we should not expect, and I do not expect to see innovations in sample prep trickling down from clinical like they did in terms of detection technologies. And I will close with one last slide. Hopefully I didn't go too far over. What I'm calling my 3 laws of rapid methods in food pathogen diagnostics. So my first law here is that there is always a trade-off between speed and sensitivity. So the probability of false negatives from any given rapid method will increase proportionally with the overall assay speed. So reductions, and that usually comes from reductions in enrichment times or sample prep or detection times. Okay, so what the proportion is and what that probability is may or may not be knowable. In some cases, it's fully calculatable. In others, it may not be. But it's really going to vary depending on the protocol in question and the overall time reduction you're talking about. The second law is that the volume of sample transferred to detection is the ultimate driver of method sensitivity. And it does this in 2 ways. It can increase sensitivity when you bring in more volume, but it also can increase the probability of inhibition, which could actually also decrease sensitivity. My final law, and this is, I think the test developers in the audience will appreciate this one. Next click, is that speed pays usually. So people will usually pay for faster methods, though it's never as much as you think, but they will not pay for more sensitive methods unless they are required to. And this is true even if they say they will. And my final point, just again to harp on this, is that non-confirming presumptives and false positives are not the same. And really the confusion between those two has caused a lot of headaches for a lot of people. And again, I'd encourage you to join my webinar next week talking about this topic in much more detail. Thanks, appreciate the time. And I will turn it over, I think, at this point to David.
Indeed, thank you. So now it's time to think about how do we choose a method that we want to use in the lab. Next slide, please, Doug. So the kinds of things that we think about are what are we trying to detect, the target, what's the method performance, how quick is it? There's a long list of things to consider. Some of those are not actually about the method itself, whether the customer will accept the results from your method is something on your customer, but the method performance itself is indeed something that we can look at. Click, please. So the target, the method performance, and the method speed are all elements that get covered in validation. Validation is a formal process. It looks at the method's sensitivity, its accuracy, what it will detect, what it will not detect, even when it's present as a kind of confusing or confounding element, how long the incubation time is, how reliable the method is, and very importantly, what matrices do we know the method will work for? Next slide, please. In the US, validations very much are the business of AOAC, and one might think, well, I can just go to AOAC, find a validated rapid method, and I'm good to go. Unfortunately, when you try to do that, you go search the AOAC site, which I did middle of August, we find there are 284 rapid microbiological methods listed on that site, including toxin methods. A couple of those are actually mischaracterized allergen methods, but ignoring that, we get down to 224 rapid methods for foodborne pathogens. So it's a lot. And in the survey, you said your pathogens of greatest concern were Salmonella, of which there are 55, and Listeria, including mono and species, of which there are 73 methods listed. So it hasn't helped us very much. And there are 50 different manufacturers represented on that list. So even, you may have a preference for a manufacturer, but there's a lot of those to choose from. Next slide, please, Doug. The methods in that long list cover a range of technologies. Dan went into it in great detail with the tiny eye chart. I've got less information, so the font is bigger, but we're thinking again about the same three broad groups: the affinity type assays, where you're capturing the target with something like an antibody, the sort of automated culture-based methods, where the culture goes on in a pot of some kind with a sensor, so that the machine detects when some threshold has been crossed, and the molecular amplification assays, which are DNA and RNA detection methods. And as we go down that list from affinity to culture to molecular, we find that we're getting newer in time, and then there are a few kind of others that don't fit in that classification. So you may have a preference for a technology. And if you don't, then how else can we think about this? Well, sensitivity was something that you raised in the survey. So if we move to the next slide, please, Doug, we'll find something which is actually very difficult to get comparative information on, which is what's the relative sensitivity of the different methods. As we look at the older affinity methods, we find that they will detect in the region of medium to high 10 to the 4, up to 10 to the 5. So in the region of 50 to 100,000 CFU colony forming units per ml of enrichment at the point that we go to detection. As we come through into the PCR layer of the table, we're down at solidly at around about 10,000 CFU per ml. If we come to a slightly newer DNA detection technology, loop-mediated amplification, our internal data says we can detect it around 10 to the 3 CFU per ml of enrichment. So if sensitivity was all that mattered, maybe we go with the LAMP system. But as always in these stories, there's a twist. So if you go to the next slide, please, Doug. And validation rears its head again. The methods that are validated are compared against a reference method. Top of the slide is the FDA BAM method for Salmonella. All of the other methods on these slides are rapid Salmonella methods. So we believe that the BAM method can find 1 CFU of Salmonella per test portion, no matter how big the test portion is. As we look down the slide, that left-hand column in the red box, we'll find that the rapid methods are all basically bracketing 1 cell or 1 CFU per test portion. Well, there are 2 reasons for that. One is that because they're being compared against the BAM reference method, and the other is because I would submit that if you have less than 1 whole cell, it's not alive. So you're never in practice going to detect less than 1 cell. If you look at the right-hand column there, the furthest right-hand column, you see that in effect what then happens is as we go down the table, we go from 100-hour or so detection by culture to a couple of days by an antibody method to a day or less than a day as we go through the molecular methods. And practically what's happened is that developers have traded sensitivity for reduced enrichment time and therefore a faster time to result. Next slide, please. And we see there time to result longest from culture, then the affinity methods, then the amplification methods, and driven primarily by enrichment time. Next slide, please. So we're pushing for speed. I actually watched Formula 1 at the weekend, and what you see there is the cars go very, very fast, and it's great while they stay on the track. If you push too hard for speed, bad things can happen. So what we're doing here is essentially shortening the enrichment time to get a faster result. That means that at the point we go to target detection, there is less target present, and we start to run up against method sensitivity. In motor racing, you can involve 1 or a small number of cars, and it can cost you a lot of money. In food and pathogens, it can involve a few to a very large number of people. People can get sick. That can come back to bite us in terms of brand value. So that's just the caution as we talk about the need for speed. Let's move on, thank you. So the other thing to consider as we're pushing up against the limits for time is there are things that can interfere with detection. On the left-hand side of the slide, there are those things that can make it more difficult for the pathogen that we're interested in to grow to detectable numbers in the enrichment. A high population of competing organisms can steal food, can steal oxygen and things, and slow down our pathogen of interest. So within the validation process, detection against a competitive background, roughly 100 times higher than the pathogen of concern at the beginning of the process, is one of the elements that goes into validation. Antimicrobials that may be present on, for example, as a sanitizer on an environmental swab, or as a component of a food material like a spice, they can also reduce the rate of growth in the enrichment. Then on the right-hand side of the slide, we have things that can interfere at the detection level. So many of the molecular methods use a fluorescence detection system, and highly colored matrices can prevent either the UV accessing the fluorophore or the fluorescent light getting back to the detector. So highly colored matrices can be problematic, and some matrices contain, for example, high phenolic content, and that can interfere with the amplification in the PCR reaction or DNA detection reaction. All of those things are a piece of the validation for a matrix. So when we get to the end of validation, we know that we can get detection against those inhibitory effects. But it does mean that if we're going to push the limits on time, we need to check the validation of those limits. The true false negative rate, i.e. failure to detect rate of any method, is impossible to know, but we can get a proxy for that information too from the validation data. So next slide, please. Let's assume for a moment that we're going to look at validation again. I'm not quite as far through as I thought I was. AOEC is really the market leader in the US. Performance tested methods are what people submit for certification. Official methods are more complex in that they need multi-lab experience and experience over several years. So you tend not to see too many official methods for rapid methods. They tend to be PTM certified. Next slide, please. The process that AOEC uses breaks up foods into 8 different categories and a separate category of environmental samples. And then within each category, there are multiple subcategories based in the food space, for example, on pH, water activity, things like that. Next slide, please. So methods will be validated against those food categories, but when you get to a layer of detail below the category, we find things may not be as complete as we would hope, perhaps. That's kind of inevitable because these studies take a long time, they cost a lot of money, and manufacturers have a need to get methods validated and into the marketplace. If you're concerned about a particular matrix and it not showing up on the certification, it's always possible to go back to the manufacturer and ask the question, do you have validation data for this category or subcategory of matrices? And often the answer is yes, because they've done it once the method has been in the marketplace. Nevertheless, the point is it's always a good thing to check. Next slide, please. So assuming now that we have come to the view that we've got a technology that we like, it's validated for the categories of matrices that we're interested in, we just want to drop it in the lab and run it. Well, again, it's not necessarily as straightforward as that. We do want to think about a few things. If this is a pathogen method, do we have the right facilities for containment of pathogens? If you're using a third-party lab that is separate from a food manufacturing site, of course, that's already in place. If you're running an on-site lab, you might want to think very carefully about whether you want pathogens growing in that space. How complex is the method? Does it fit easily into our established lab workflows? Now, most of these rapid methods start with an upfront enrichment process. That will be very familiar to anybody running cultural methods. If, however, you're currently only running cultural methods, you may need some special training for your analysts, because there are definitely some techniques and some skills that are a little bit different running rapid methods as a class. If you're already running one platform, you'll probably find training on the method is very easy to switch to a new platform. Any method that we bring on board and start to run, of course, will need some documentation. We do need training. We do want proficiency testing. That's really not a differentiator in any sense because all methods have that requirement. Verification is an interesting thing. That's proof that the method works in your hands or in our hands. So when we start up a new method, that's about picking a validated category, validated subcategory, running some testing to show that the method in our hands gives us the results that we would expect. And then that allows us to go forward. And then fitness for purpose really relates to the specific matrices that you're concerned about. And there are ways to do that. They're not very complicated, but it's certainly worth a discussion perhaps at some future time. So if we go to the next slide, please. There are lots of rapid methods on the market. We covered that. because of the requirements of the validation systems, there's not a massive amount to choose between them in terms of sensitivity, accuracy, and precision as applied to the test portion of a matrix. What certainly differentiates them is the total time to result and the range of matrices for which they're validated. So it's the total time to result is something you can get from the validation certificate. The validated list of matrices you can get from there too. It's definitely worth ensuring that the method is appropriately validated for your matrices, which can be as simple as asking the manufacturer, might involve generating your own validation. We would, of course, strongly recommend that you use an accredited lab who can not only help with method selection, but could also help with that additional validation. And finally, of course, if you have any concerns at all about a method that you're contemplating, ask your lab or ask the method supplier. Thank you. Let's move along and Doug can finish us off.
Fantastic, David. I'm incredibly honored to be coattailing on the shoulders of giants here. So you attendees here are seeing a lot of Eurofins microbiology firepower at the table today. So I get to be the cleanup better. And once you've chosen the technology to do your testing, now then what else do you need to consider? Because you know, the technology works as it's designed, but you can really screw up your testing if you don't follow the next section. So this is a pretty important section for you to think about. So here are some practical test considerations. So when you're thinking about the materials that you're going to be testing, you know, you have a lot of choices to make. So if you're doing supplier verification, you're looking at your raw materials and your ingredients. If you're looking at in-process or finished product testing, you're probably dealing with a more complicated matrix than just a simple ingredient. And then on the environmental monitoring side, are you testing dirty surfaces? Are you testing recently cleaned and sanitized surfaces that have puddles of sanitizer residue remaining on surfaces? So all of these choices on the sample that you're testing can have a huge impact on whether a technology is able to detect a pathogen or not. The other thing you have to consider too is all the practical consideration of your test program. So if you're doing very infrequent testing, that's going to leave gaps of knowledge in your food safety plan that might open up the door to very large recalls. And that's one of the advantages of doing multiple testings and high frequency testing, is it reduces your risk of large recalls. So if you can isolate the problem to a particular SKU or a particular lot number, then you're going to be saving a lot of money by saving in how much product you may potentially have to recall. How many samples do you test? We've got some International Commission recommendations on the number of samples, and we have very, very, very few customers that are even coming close to doing that kind of sample volume. So again, we all work for a testing laboratory business. We're biased. We'd like to have you send us in more samples, but to do really valid sampling plans, particularly for pathogens, when you're really not expecting to find pathogens, if your food safety program is working. So if you're only doing one small 25-gram grab sample out of a large production lot, and you're making a ship or no ship decision based on that analytical result, all that result is telling you is what's present in that 25-gram sample. It does not necessarily tell you what's in the entire production lot. What sample size do you collect? You know, it used to be you collected 1 gram and you did a 1-gram pathogen test. Then we realized that that was not sensitive enough, so we went up to 25 grams. Then if you're following FDA's Salmonella sampling guide, now you're up to 375, 750, 1.5 kilogram sample sizes, because the whole goal here is to try and find that pathogen in that production lot so that you're not shipping contaminated product out into the marketplace. What are you testing for? You know, a lot of people say, well, we don't have to do pathogen testing because we're doing indicator testing. Well, that's fantastic, but you better have some validation data showing that the population level or the presence of your indicator has some relationship with the prevalence of the pathogen in those specimens. And then you're setting performance specifications based on what you're testing for. Do you have very stringent high expectation performance specifications? Or do you set your specifications so that you look good all the time, so that you have no drama in your company because you're always meeting these specifications that really aren't very stringent? You need to question whether or not it makes sense to do pathogen testing in-house, as David alluded to, because you're going to be cultivating large populations of pathogens. Even if you're doing indicator testing, the odds are you will be growing pathogens in that in-house laboratory. This is a biosecurity threat for the facility, and this is one reason why many companies decide to outsource their pathogen testing, because they don't want to have gigantic populations of pathogens anywhere near a production floor. Again, speed is the key. There are many platforms out there that will tell you that we've got the most quick test in the marketplace, but what they don't tell you is what the sensitivity is. So as David also mentioned, and Dan also alluded to this, and that is the quicker test result usually means the reduced level of sensitivity of the tests. How do you manage false positives and false negatives, or non-confirming positives? So we've discussed that. And then if you get a presumptive that doesn't confirm, what should you do? And of course, our posturing is you should treat that as if it is a true positive and move quickly to resolve your issue. Okay, back to the question of what do you test for? Again, there are many, many indicators that may have value in your facilities, and Eurofins can help you make those decisions on what is appropriate. I've gone into many companies that are having pathogen detection problems, and one of the root causes is they're only using ATP for cleaning and sanitation verification. ATP may be useful to verify the cleanliness of surfaces, but it may be poorly correlated with the prevalence of pathogens. And so it was just simply using a test that was not fit for purpose to answer the question, do I have pathogens under control or do I not? Again, a lot of times folks are focused only on salmonella or listeria testing, and they make the presumption that if I can control these two pathogens, then the odds are good I'm controlling the other foodborne pathogens that might be in products or in environments. So that may or may not be true with your product and process combination, but if you are ignoring the others, you might want to have some sort of verification that you are also controlling the other pathogens that might be high risk for your product process combination. Okay, product sample considerations. You can get increased sensitivity if you are testing liquid samples, because you could maybe direct plate if you're doing a standard culture kind of pathogen detection. And that might give you a lower limit of quantification or limit of detection, depending what kind of test you're performing. Solid samples will require an initial dilution, which, because of that dilution, you will get reduced sensitivity unless you test the entire portion of the diluent. Powders have the potential to create dust, which is a contamination risk for the laboratory. And you should ask your laboratory what kinds of controls do they have during sample manipulation to prevent these aerosolation of powders into the facility. And then, as David said, some ingredients contain inhibitors for the particular biochemistries that might be used for detection. So when you're thinking about, I've produced a production lot, I need a verification data point that my food safety plan was working. How do I gather that data point? What do I target? Well, if you're just pulling a single grab sample from an entire production lot, I would ask the question, is that adequate? Now, for liquid samples where you can get constant homogenation and you pull that sample from that large production lot, I think you could argue that that small sample is representative of the entire production lot. But if you've got solid samples that are broken into smaller primary packages that then may be in larger boxes that are secondary packages, or put in palletized containers or something like a big super sack, then where do you collect your representative sample? Is it on the top? Is it on the bottom? And is it in the middle? Is it the beginning of a production run, the middle of a production run, the end of the production run? All of these questions you need an answer for. You needed to justify why you collected the sample that you have. The labs, they don't care about the sample. They assume that whoever collected the sample, that the sample is uniformly representative of the entire production line. And that may or may not be true. And so when people blame the lab for a bad result, be careful, because it may be nothing more than a reflection of a poor sampling plan by the people who were collecting the samples. We have some guidance then on sample, the number of samples to take. And if you look at FDA's salmonella sampling guidelines, they categorize foods in 3 categories. So a category 3 food is the lowest risk. The recommendation is to collect 15 25-gram samples per lot. So again, that's pulling 15 individual samples. Each one of those is 25 grams. Okay. A food that is ready to eat, but it is destined for an average consumer, then the recommendation is 30 25-gram samples. And then if it's a category 1 food, so this is a product that is ready to eat, destined for a high-risk consumer. So hospitals, daycare centers, congregate care centers, and the like, then we're up to 60 25-gram samples. So let's think about these sampling sizes and just do a reality check. There is one report that demonstrated an infectious dose for salmonella in cheddar cheese was 1 colony farming unit in 500 grams. So if you are only collecting a 25-gram sample, or maybe you're compositing up to a 375-gram sample, what are the odds that you'll be able to detect 1 cell of salmonella in a 500-gram portion of a production lot? And so the odds are relatively low at a 25-gram sample. So you would have to test a whole bunch of these before you even remotely came close to being able to find 1 CFU per 500 grams. And you go back to Dan's earlier slide about finding a needle in a haystack. That is the issue with both sampling size and the number of samples you collect. For environmental monitoring, what do you test for? So in wet processing environments, the usual recommendation is target Listeria monocytogenes as the pathogen of greatest concern. And the reason is because this pathogen can thrive and multiply and grow in wet food products and in wet processing environments. And we typically will do testing using a broader indicator than just going directly for the pathogen. And so we're testing for Listeria species. In the dry processing environments, again, depending on the product and the intended consumer, most of the time we're targeting salmonella. If it's powdered proteins or powdered formula, we may also be testing for Chronobacter, because that's going to a high-risk consumer group. And the indicators that are promoted for testing dry environments may be Enterobacteriaceae, because some people believe that it is the highest correlated indicator for the prevalence of salmonella. That may or may not be true in your product process combination. Or sometimes you could do coliform E. coli counts as a verification of cleaning and sanitation. So what about the other pathogens? Again, the argument is if you're doing a great job controlling Listeria in wet environments, great job of controlling salmonella in dry environments, then the odds are good you'll be controlling the other pathogens in those similar environments. Let's talk about transient versus persistent strains. So there is a religious belief out there in the field of food safety that you should never test your highest risk direct product contact surfaces for pathogens. And the argument is if you find pathogen on those surfaces, then the product that was recently processed on those surfaces is potentially also contaminated, resulting in a recall event. But I would argue if you're doing sanitation verification, then what is your risk that you're going to find pathogens on those surfaces? I mean, if you don't trust your sanitation, you've got bigger problems than zone 1 testing for pathogens. But let's just assume that you decide to follow that religion. So if you do find a pathogen on a surface, another argument that you have to resolve is, did that pathogen just float in on that surface after cleaning and sanitation, land on that surface before I took the environmental sample from that surface? So this can happen, particularly if you're processing products where you're bringing in pathogens into the facility, because it's present on raw produce, it's present on other raw agricultural commodities, it's present on raw meats, poultry, and seafood. So if it's coming into the plant, it could occasionally get on a recently cleaned and sanitized surface. And if you test that surface, you occasionally might find it. Versus a persistent strain, this is a strain that is to establish residence in your facility. If it establishes residence on your direct product contact zone 1 surfaces, and you never test those surfaces, how will you know whether you've got a persistent strain on that equipment near product contact surfaces? And we've had some very, very high profile outbreaks because companies were doing relatively decent environmental testing on non-contact surfaces, but they were following this religion. They didn't know that they were routinely adding persistent resident strains of pathogens into product, and they weren't detecting it through in-product testing. So you really need to know, is it transient or is it persistent? And if it's transient, then you need to also understand why your control programs are failing to eliminate the harborage site for these pathogens. How many environmental monitoring samples should you take? Again, depends on what your appetite for risk is, but if you really want to protect your brand, then taking a handful of samples once a week may or may not be a sufficient number of samples to take at a sufficient frequency to legitimately protect your brand. So I can give you maybe some examples. So when FDA or a local regulatory inspector comes in and does a super swabbing event, they're going to be collecting 100, 150, maybe 200 samples when they're doing that investigational monitoring, because they're really trying to find out whether you have pathogen in your environment or not. So if you're only doing 5 a week and FDA is doing 100 or 200 on a particular investigation, that's a pretty substantial gap between what you're doing versus what a regulatory inspector does. So again, the table on the right, those bullet points just give you an example of a recommendation from a dairy trade organization. Depends on the size and complexity of your facility, depends on your product process combination, and which pathogens are of greatest concern for your environmental monitoring control. So we have concluded the formal presentation. I am pleased to tell you we have just a few minutes to try and address some questions. So Amy, let's drop it back in your court and see where we go next. Thank you very much.
Wonderful. Thank you so much to our presenters. I am just going to go ahead and launch the poll here. Again, just asking if you would like someone to follow up with you after the webinar. So again, I'll just give a few moments here and we will move into the Q&A. So thank you to everyone who has submitted your questions. The first one that has come through, the question here, several companies now offer PCR or real-time PCR systems. How does one choose between them?
Sounds like a Dan question.
Yes, it does.
Sorry, I was on mute. I knew you were going to send that to me for some reason. How does one choose between them? Well, that's a very difficult, a very interesting question, and I guess there's lots of ways you can look at it in terms of performance. I mean, they're very similar, right? They've all got very similar sort of levels of validations and verifications, and you have some systems that have been around, platforms that have been around much longer than others. You have, and that doesn't necessarily mean they're better or not, but it does mean they have a longer track record at least, or potentially at least more exposure to more in different matrices and sort of real world scenarios. But again, that doesn't necessarily mean that they're better. It's really difficult. It's a really difficult question, and you can say, well, let's look at price, right? Let's go with the lowest cost option. Again, may or may not be the right decision, just like as in the real world when you're making purchasing decisions. Price is probably it's a factor certainly that you want to consider, but it really shouldn't be the deciding factor, particularly for something this important. I think what I would suggest is you have some comfort level with the company that you're going to deal with. Make sure that they are, not just a flash in the pan, that they have, they have a dedicated customer service. They've been around. You understand that you can work with them. They can work with you. And they're going to be in it for the long haul to support you, because it's not just easy that I buy this PCR platform and I walk away. You are going to need support from them, and just like you're going to need support from your testing lab if you use a testing lab. So that relationship's important. If you have those personal relationships, that's probably the best way to make those kind of decisions.
Great, thank you for that.
Disagree with any of those points or want to add anything? Please do.
I think you nailed it.
Okay, great. Next question here. How can we shorten turnaround time when we have to retest?
David, do you want me to take that one?
It's either Doug or me. I mean, I think the question would be, why are we retesting? And Doug can probably back this up, but the original test result doesn't ever go away unless there is some very clear and very definable reason why there was a problem or an error in the first result. So we've tested a sample. We've got a pathogen detection in a 25-gram sample, and as Doug's example, the one cell in 500 grams. So if we go do a retest, it will almost certainly be negative, but it doesn't mean to say that the pathogen isn't there. So I think the first thing is, as Doug said, if you get a detection, you have to treat it as a real detection unless there's a very clear, definable problem with the test. So my feeling would be that retesting isn't something that needs to be done quickly. It isn't necessarily something that would be done at all, certainly with the intention of overturning the original result. Doug, do you want to add more to that?
Yeah, there are some things that the laboratory just simply has no control over. Number one is we can't change the biology of the target that we're dealing with. So if we have a portion of the original specimen that was submitted, which we should, right? So we'll get a larger sample volume than we really need. We have a portion of that in retain, then we can immediately go and enrich that portion and begin the testing again. So the laboratory may not have to wait for the customer to resample and ship in that new portion. So that's one way in which strategically we can shorten the retest time. Another thing that the laboratories may do that may not be apparent to the client is if there's some issue, some quality issue, or the analytical result just doesn't make sense with the initial detection run, then the laboratory could potentially go back to the original enrichment and run a new detection. So again, the client probably would not be aware that that was even being done, and that would have just a small increment in extending the turnaround time to a result, is that the lab has to do retesting of an enrichment. But to David's point, retesting can never be used to disregard a prior positive, unless the laboratory has a quality control failure and they cannot stand by that original detection result.
Great, thank you. And it looks like we have time for about one more question. So the last one here, how do I validate the method or extend the validation if I have a matrix not covered by the AOAC certificate?
Would you take that as well, Doug?
Go ahead.
So essentially, if you go online and do an online search for AOAC Appendix J, that will tell you everything that you need to know about how to run method validations. That document is set up for people that are method developers, generating data to submit to AOAC for certification. But the process is fundamentally the same. So if you want to get a AOAC style validation for a matrix that hasn't been validated at all, you'll be testing nominally 30 samples, 5 that are not inoculated, 5 that are inoculated at what's called a high level, and 20 that are inoculated at a low level designed to give very broadly 50% of those samples testing positive. You run that by the rapid method and the reference method alongside one another, compare the results, and if they give you the same detections, your validation is done. It's a little bit more complicated in practice because hitting those exact fractional levels is tricky, so you probably end up doing more than 30 samples, but broadly speaking, that's the approach. Now, if the overall matrix has been validated, but you're in a category that's adjacent, you might not need quite so much work. But it's basically all there in Appendix J.
Right, thank you. And I just wanted to remind everyone, if that we didn't get an opportunity to answer your question, we will respond offline. And I know we've had several other questions come in, and just a couple reminders that you will receive a copy of this recording, as well as a copy of the slides within 3 business days. So again, we will definitely send that out to you. And I would like to thank Dan, Doug, and David for your presentation today. Thank you again, everyone, for attending, and I wish you a great afternoon.
Thanks, Amy. Hey, so I never got to see the chat box. Were there a lot of trues or versus falses for my question, my true false question? Can